Satiko, thank you. I want to first thank Gert Covenberg for helping arrange this seminar with our very distinguished visitor, Dr. Omer Bayraktar. Omer, I've known Omer now for a few years and through Welcome Leap. And every time I see him, I come away inspired, and he's got tremendous amount of insights into a large number of things. And so give you a very quick intro to Omer. Some of you know Omer. Yeah, he got his doctorate degree from University of Oregon, working in Chris Do's lab. Chris Do's HHMI investigator. Then he went to David Rovich's lab, University of Cambridge, as a postdoctoral fellow. He was there for a couple of years, few years as a postdoc. Then in 2018, he's appointed as a group leader in cellular genetics. His work has always been in the brain. He started with fly brain, and he discovered something very, very exciting in the fly brain. Then he went on to human brain, and then he was one of the early ones to map in situ the cellular heterogeneity of the human brain. Since then, he's done really tremendous work. He's one of the hopes for glioblastoma. He has mapped the trajectory in human glioblastoma. He works on autism. He works on developmental issues in human brain, fetal brain. And then, in fact, I'm really privileged and delighted to have communications and conversations with Omer. And Omer, I really am looking forward. We are all looking forward to your talk. So thank you for coming and visiting us here.
Thank you so much. I think the inspiration actually works the other way around. I'm flattered and embarrassed to get such a generous introduction from you. Well, thank you all so much for coming. So yeah, it's really my pleasure to be able to tell you about what we do in my lab. We are interested in human brain disorders. We're interested in discovering the biology of these disorders using single-cell spatial genomics tool. So I'm really looking forward to telling you guys what we are up to. So we get started here. Let me get rid of that before. So there's really two simple reasons that we focus on human brain disorders. First, there's just incredible unmet clinical need. As most of you are aware, many brain diseases such as dementia or high-grade brain cancer are incurable. And we really need to think about how do we make things better for people? How do we make things better for patients? But the second, as sort of a group of fundamental biologists, I think we can easily trace this unmet clinical need to our inability to understand the biological mechanisms that underpin these diseases. So to make it a bit more catchy, I like referring to these biological mechanisms of disease as rules. And the example I picked for you here in this slide is from high-grade brain cancer. So as many of you may know, in these brain cancers, cancer cells are not a monolith. They can exist across a spectrum of different cellular states. And in high-grade brain cancer, the prevailing model of how cancer cells transition across these different states is what I describe as the chaos model. It's really widely thought that cancer cells have this unrestrained plasticity that enables them to zip around across all these different states and evade treatment. And obviously, at some level, this is an exaggeration of cancer genomics. We know that these tumor cells have a lot of bad, genetics. They have a lot of mutations, and that sort of just gets them out of control. But what it's led to is really almost this nihilism in this field that, that the biology is so complex, so intractable that we're never going to be able to fix it. However, what you can imagine here is that there could be an alternative. And an alternative could be that while these cells are still aberrant in their biology, they actually obey rules, right? They can still transition across different states, but they can actually follow a stereotyped trajectory that goes from A to B to C. So really our long-term interest is to be able to identify such rules. And if we could identify these, we really believe that we can then go about twisting these rules and develop targeted treatments. Right. So, we think a lot about why haven't we able to sort of move from the chaos model into the rules model. And most of the time, people will tell you that it's stuff like the heterogeneity of all the different cells in the disease brain that's the problem. We actually think, while heterogeneity is a problem, it's more of an excuse. The real problem is that we don't really understand the fundamental human biology of these diseases. So we could break this down into a couple different things. Number one, There are lots of cellular and animal models of these diseases that are widely studied, but unfortunately, they don't really recapitulate key aspects of pathology, right? The stuff that works in mice, that can cure brain cancer in mice, does not really work on people. Number two, sort of a key knowledge gap is we don't really understand what diseased human cells look like in the patient's brain, right? So we just, again, a lot of our knowledge and information goes back to certain animal models. And number three, sometimes we actually know kind of the genetic causes of these diseases quite well. Like in cancer, we can map out all the driver mutations and such, but we know very little about how these specific disease perturbations link to these cellular and the tissue phenotypes, right? And as you know, as you heard through Shankar's beautiful introduction, my background is none of this human disease stuff, right? I'm just a developmental biologist who first worked in fruit flies, then in mice. But I really want to be able to bring the perspective that we built in the model organisms, where we can answer these two questions. We know what the cell types are in the fruit fly brain, in the mouse brain, and we know if we make a mechanistic perturbation, if we knock out a gene, we can easily link it to a cellular and a tissue phenotype. So I really came into Sanger, a genomics institute, in 2018 with the hopes that the things that taught us the mechanisms of brain development, we could basically apply the same kind of tricks to crack the mechanisms of human brain diseases. And really, when I started out, there were really two fundamental reasons why I joined a genomics institute. I wanted to be able to use first single-cell genomic technologies, right, which allows you to take a complex tissue, break it down to identify, the disease-vulnerable cell types in their molecular programs. But number two, use this relatively more recent crop of technologies that are called spatial genomics, where we can do this kind of stuff we're doing with single cell in the in situ spatial resolution. So I think, again, I'm preaching a bit to the choir. I think you guys are very familiar and you're on the vanguard of some of these technologies. But I think the overall in neuroscience community, the community at large agrees with this as well. And I was really lucky to be part of this big consortium that put out these three kind of like review and community consensus pieces in Nature Neuroscience last year on the values and still sort of the challenges to the use of these technologies for discovering brain biology. So as you also know, we do a lot of stuff with spatial omics. I think, again, a lot of you know that single-cell omics, you get the list of parts. Spatial omics allows you to put it together, look at how different cell types are interacting in tissue. But really what I'll try to demonstrate to you today is that in our research, in many other areas of research, spatial is not just nice to have, it's actually essential to have. It really allows you to interpret the biology of the cell types. So really, when I started in 2018, single-cell omics was already a big thing in Sanger. This is the beginning of the human cell atlas there. I was about to just hit its stride. But the task I set to myself was to actually establish large-scale spatial omics there. And the way we kind of did go about doing this is, back in the day, we were mainly relying on measuring just a few genes with technologies like RNA-scope single molecule fluorescent in situ hybridization. So this is like a mouthful, but it's basically SM-FISH. It allowed you with really, really exquisite sensitivity to measure single cell expression of generally up to like 4 genes. And this is a lot of work that my first two postdocs, Kenny and Tong, did in the lab. They're able to set this up, using sort of continuing the stuff I did in my postdoc. We were able to use this as an orthogonal technology to validate a lot of single cell studies of different human cell atlas projects in Sanger at that time. You can see a few examples from the fetal thymus and endometrium. But we always wanted to go beyond that. We always wanted to be able to measure the full transcriptomes in situ. And really the step change came with the commercialization of a spatial RNA sequencing technology called Visium in 2019. Again, many of you guys are familiar with it, but Visium is basically a very fancy microarray technology that allows you to positionally capture transcripts from tissue and read them out with NGS. So you can basically end up with really this kind of situation where you get the full transcriptomic readouts of tissue locations all over your sample, right? However, in 2019, there's a big limitation, right? This Visium microarrays did not have single-cell resolution when they first came out. And still the most dominant, widely used form of this technology is about 55 microns in diameter, each of those microarray spots, which means in most tissues, they profile the transcripts of multiple adjacent cells, right? So back in the day, to be able to deal with this, we actually pivoted into computational biology. We thought that this is still great data, but maybe we could computationally extract cell type specific information from here. And it was really around that time we started this really incredibly rewarding and ongoing collaboration with Oli Stegel, who was at the European Bioinformatics Institute, EBI, then, and is now for a number of years, has been set up in Heidelberg at the German Cancer Research Center in Embl, a lot of affiliations. And our shared graduate student, Vitaly Kleshchennikov, what he realized was that we could go about doing this if we integrated spatial Visium RNA sequencing with single-cell RNA-seq. So to be able to do this, he put together a probabilistic computational algorithm that we call saltolocation. And what saltolocation does really is that it uses the single-cell transcriptomics as a gene expression reference to be able to go into Visium or any type of spatial RNA sequencing data and deconvolve cell types, right? So the example here, which was published in the paper, is from the mouse brain. So what you're going to see here at the beginning, we're looking at a single nuclei sequencing data set of a mouse brain. And you got your UMAP embedding. These are all different cell types. And you can see we have sort of picked 6 different cell types here. As I play that, you'll see we're going to bring in a Visium spatial transcriptomic data set in the mouse brain. And what saltolocation does is literally it goes into each one of these 55 micron locations and infers the abundance of every single cell type mapped out or defined in this single cell transcriptomics. So, you know, when we came up with this, I thought it was quite neat, because it allowed you in one step spatially map all the cell types you defined in single-cell RNA sequencing. So if you're kind of used to the old school practices of you're looking at 4 genes at a time, you have to do lots of different panels to cover all different cell types, this really addresses that type of limitation. But what I really did not anticipate when we were doing this and pre-printing the tool in 2020 is how widely it would end up getting used, right? So starting with many of our collaborations with other human cell atlas groups at the Sanger Institute, but just going beyond us widely into community, saltolocation, I'm proud to say, has now been used to map every single human tissue. And beyond healthy human tissues, it's been really extended to a vast array of different diseases. Some of my favorite applications are from Julio Saez Rodriguez's lab, now in ABI. They really applied it beautifully to human myocardial infarctions. And some of the more exotic applications are in the salamander brain regeneration, which is a wonderful paper from Barbara Chartline and I think L.A. Tanaka back then. So saltolocation, we still use it. You'll see quite a bit of data from brain cancers when I present that. But, you know, as the spatial technologies evolved, we wanted to stay on top of the times. And we were quite interested to go into the realm of spatial technologies that allow you to measure many, many genes, not just 4 genes, at true single-cell resolution. So to be able to do this, around 2019, I teamed up with Sarah Teichmann and John Marioni at Sanger and ABI, respectively, at that time. And we started an internal initiative called the High Throughput Spatial Genomic Initiative. Again, mouthful, HTSG for short. And what we did through HTSG was to really introduce these new crop of imaging-based spatial transcriptomic technologies to Sanger. There are lots of different types of these technologies. The one we picked is called in situ sequencing, which works really well in human tissue samples. And again, I think a lot of you are familiar with it. The idea here is that through probe-based targeting and really clever cyclic imaging and barcoding, you're able to really image single cells in the tissue, and you're able to basically pinpoint individual micron-sized RNA molecules as these spots. So at the end of the day, you could basically segment the cells from the images. You can count how many spots of each transcript are expressed in which cell, and you can use that, literally, you can analyze that like you analyze single-cell transcriptomics. And again, using that, we were able to, for example, create maps of the developing human hindlimb or lobe limb. And what you're seeing here, using this type of technology, we're able to image single cell types across the entire tissue, and we're able to find different types of bone, muscle, skin, so on and so forth, all the different constituent cell types that make up the limb. And really skipping ahead dramatically, with the help of very many, again, I'm very proud to say that at Sanger now, we have one of the highest throughput spatial genomic pipelines. Together, we built a pipeline called the Spatial Genomics Platform. And the crown jewel of this is the commercialized form of in situ sequencing called Xenium from 10x Genomics. We have a fleet of 5 of these instruments. It's located in the coarse pipelines of Sanger, so it's no longer just sequencers. You can submit your tissue samples there. What they will return to you is single cell measurements of several hundred to up to 5,000 genes on the same sample. And really what we've built, again, with the contributions of many others, is a fully automated high throughput pipeline. And that has enabled us, in the span of just little over a year, profile over 155 million cells from across different human tissues, over 30 different human tissues and diseases. And a great deal of this is the highest plex version of the technology, 5,000 plex. So you get a lot of data that is approaching near single cell transcriptomic quality, but also with the spatial resolution. So I'm still quite interested in different new spatial technologies. We're always on the lookout what to bring to Sanger, what we can scale, but I kind of want to move on to the title of my talk, which is, what do we do with all these tools, all this like wet and dry lab approaches? So in our lab, as I think I've told 15 different people today, we're really focused mainly on cell atlasing of the diseased, human nervous system. About 60% of our work is on adult brain cancer, 30% is on neurodegeneration, and about 10 or 20% is on neurodegeneration, and about 30% there, and about 10% in neurodevelopmental disorders. And today, what I'll tell you is really, I'm going to try to cover what we do for neurodevelopmental disorders really briefly, share some unpublished work with you. Then I'm going to review some of our recently pre-printed findings in adult brain cancer. So before I lay out the big questions we have in neurodevelopmental disorders, I want to share with you, or I want to remind you that the disorder that we study, autism, is quite complex and diverse, right? So when you think of autism, it's a spectrum which has lots of different sort of cognitive phenotypes and genetic sort of causes. But the type of autism that we focus on is what you could describe as severe autism, where it is highly disabling and debilitating, right? So this really manifests itself more as developmental delay and intellectual disability. People with severe autism have, could be non or minimally verbal, and they have really really difficult conditions like, persistent epilepsy, so on and so forth. And by some estimates, about almost up to 20% of autism cases in the US and beyond represents this. And this also turns out to be the form of autism that we understand genetically the best. And what we know, at least 10% of the risk here is conferred by rare coding variants in individual genes, right? So these are variants that appear in the protein coding regions of these genes. And if you have a single one of these variants, which could be, leads to loss of function of that allele, you have a pretty high, disproportionately high risk of developing severe autism. Then really the big question that the lab is chasing in this area is, what are the cell types and the regional brain circuits that are affected by severe autism in the brain? And really the challenge that we are facing here, I think, is twofold. First, there are lots of genes with these rare coding variants. Again, this is kind of the part of autism genetics that the community agrees on the best. So we can really come up with a list of 300 genes, everybody's favorite genes that have these risk coding genes. That's a lot. But second, as you all know, in different parts of your brain, there are thousands and thousands of different cell types. So to be able to study what 300 genes do across thousands of different regional circuits is a huge challenge. So really what the field and our lab is chasing right now is to understand whether there's convergence of these genes. Are there particular cell types, particular brain circuits where all these different variant, carrying genes are actually doing stuff for during development? And again, this is a problem that has been addressed by others. The way most people go about this is that, they basically track where the genes carrying these variants are expressed during normal brain development. Right, so they could tap into bulk or single-cell transcriptomic studies of the brain across different developmental stages into adulthood. And again, this has been done by many others in really beautiful ways. If you were to sort of grab your 300 genes, overlap it with this risk of genes, you can then say, where are they enriched? Where are they actually converging? And really kind of what the field agrees, again, is that these rare coding variant carrying genes, which, to just make it short, I'm going to be referring to from now on as autism risk genes or autism susceptibility genes, seem to converge in the human cerebral cortex, specifically around mid-gestation And it's thought to be also in upper or deep cortical layer excitatory neurons, depending on who you ask. So this has been around a while, and we thought that this required a revisit, right? Because we know that autism phenotypes clinically are multifaceted. So it was not really crazy to think that different brain regions could be involved. But second, a lot of this stuff was basically just simply single cell or bulk RNA-seq. So we thought it was really timely to validate these really important findings with orthogonal technologies. So about a couple of years ago, with a small director's award from Simon's Foundation, from the Safari program, with our wonderful longtime collaborator, Tom Novakovsky from UCSF, we went about creating a single-cell spatial resolution of autism risk gene expression during normal human brain development. And really, I just want to walk you through this slide before we go into the data. So the first thing we did is that we grabbed first 250 of these autism risk, autism susceptibility genes. Again, these are the genes that carry these rare coding variants. Then we coupled them with a bunch of different cell type markers of developing human brain, progenitors, differentiating neurons, glial cells, so on and so forth. We focused specifically in mid-gestation, so about 16 to 21 gestation weeks. We looked into brain tissue from 6 different individuals. So these are basically fetal brain tissue that's collected for research in the UK for elective terminations. So there are no known conditions, no known comorbidities. And obviously, what we're looking at here, as far as we can tell, is normal human brain development, meaning that we haven't, genome sequenced these fetuses to map out whether they have an autism risk gene variant or so on and so forth. But what we were able to do using the Xenium technology, we were able to go into these samples, and what you're seeing in the middle here is a DAPI stain of a big tissue section, which is almost 1 by 2 centimeters. And again, as you saw before with Xenium, we are able to really measure the expression of all of these 250 risk genes at single cell resolution across the entire sample. And once you do that, we can then use anatomical landmarks to assign the cell types into these different brain regions. Right. So when you do this and throw all the data you have from this one donor that you're seeing here into a UMAP embedding and color that embedding by the anatomical regions of the cells, the first thing that you see is that cells are clustering according to their regional identities. Given that most of the genes we're measuring here are autism risk genes, that tells you, that's your first clue, that autism risk gene is actual expression is very different across all these different brain regions, right? However, we really wanted to be able to do this in a much more kind of like unbiased way. And for this, what we did was we took all the data that we had generated, which amounts to a little over 10 million single cells from 6 donors from almost up to, I think, 15 different tissue sections. And what we used was an analysis approach described as non-negative matrix factorization, which allows us to identify autism risk genes with correlated gene expression. So we put all the cell type markers away, we focus on these 250 risk genes, and we see if these guys converge with one another in single cells from different brain regions. And when we did that, we found that a really small number of factors, which are just basically correlated gene programs, express the convergence of these autism risk genes. So you might look at this, you might say, what is NMF? What is this heat map? This doesn't make sense. But given that we're actually doing this in the spatial data, we're easily able to investigate what these programs mean. So if we were to grab one of these programs, which is anatomically really enriched, as you can see in this map, part of the brain called the germinal zones. These are the parts of the brain that the neural progenitors are enriched. You're able to see, if you were to sort of focus on the cortex, where we know the anatomy and the, of the developing cortex really well. We know that the stem cells are near the bottom, and there's a lot of them at this ventricular surface. And going up there in the middle image, that's where you have your differentiating neurons. We could really look at this as single cell resolution, which is what's shown here. Every dot is a single cell. And we could really measure that indeed the genes that make up this program are enriched in expression in these stem cell zones over the cortical plate with the differentiating neurons. Right. So again, we could do this for each one of those 5 different factors, 5 different programs. We can do this across multiple different donors. And what we see is like this really consistent picture of autism risk genes converging in a small numbers of regionally distinct programs. So when we did this, I thought it was, again, quite neat in that, right, we have a small number of, regional gene expression programs that we could go about explaining. But really, my initial expectation was that in terms of the risk genes that contribute to these programs, most of the genes would fall into these sort of like cortical development focused programs. So I expected most of them would be maybe in the cortical plate, which a lot of the previous studies said. A bunch of them could be in the germinal zones and part of the brain called the subplate. What was really, really striking to us was that if we asked the number of genes that associate with most of these programs, the number one region that had the highest number of risk gene expressed was not the cortex, it was a part of the brain called the thalamus, right? So before I tell you about what the thalamus does, I just want to sort of show you that this is not just because of some core key aspect of NMF analysis that we came up with this. If I now pick the risk genes, including some very well-studied risk genes like SN2A, right, which is an ion channel, and if I simply show you the in situ hybridization of these individual genes, you see them light up the part of the brain that's the thalamus. And indeed, if we kind of zoom into the thalamus and compare it to the cortex, and if you look at where the RNA transcripts marking this gene are, you indeed see that these genes express in the thalamus. And furthermore, if we simply just now throw away all the fancy analysis, we're just looking at the expression of autism genes in cells across different anatomical regions, we see that many of the risk genes show elevated expression in the thalamus over the cortex. So we've done a bunch more on this. We added cell type specific information onto this that showed us that in the thalamus, these genes were specifically expressed in excitatory neurons. We were able to demonstrate that the risk genes that are enriched in the thalamus over the germinal zones are more likely to be synaptic, function-related genes, whereas in the germinal zones, you get a lot of like transcription factors, gene regulation. And furthermore, within the thalamus, there are different subdivisions of that structure. We were able to demonstrate the risk genes actually show kind of like variable expression across different parts of the thalamus. They're not just expressed all over. But really, the kind of like the take-home message from this is that one of the sort of like, potential hubs of autism risk in human brain development is not necessarily the developing human cortex, but it's actually the developing human thalamus. And does this make sense, right? So this to us makes a lot of sense because the thalamus is sort of like the sensory relay center of your brain. So besides except olfaction, all the different sensory inputs, so vision, smell, touch, all of it actually goes from your sensory organs to the thalamus first before it's relayed into the cortex for higher ordering processing. Then the thalamus and the cortex engage in this sort of like two-way conversation to process that sort of input and do something about it. And this all makes a lot of sense because hyper and hyposensitivity to these different sensory cues is actually a core autism symptom. That's part of it's a diagnostic symptom of autism. And on top of it, autistic individuals have known structural connectivity from neuroimaging studies in their thalamus. So I think you might forget everything about this portion of the talk. What I'd like you to kind of remember is that using this type of approach, unbiased, we're just looking at where risk genes are expressed in the brain, we're able to pinpoint a completely new brain region that in a way was not on the radar of this community. And I think what's really cool about that is now we're in the age of cellular and organoid modeling of these risk genes. Everybody's super interested from organoid models to animal models, trying to decode what the functions of these genes are looking at. And I think potentially we are asking the right questions in the wrong model systems because we're all so focused on the cortical circuits or the cortical organoids, whereas we should be actually looking into what these things are doing to the thalamus. And beyond that, I mean, I love describing this approach as a spatial transcriptomic GWAS approach. That's wrong. But I think that's a very, very cool and promising approach to think about all these different kind of like genetic study derived, like causal genes and so on and so forth to sort of contextualize their spatial temporal function. I think spatial transcriptomics is a basically great tool for that. Okay. So with that, I want to dramatically shift gears. I'm going to tell you about what we've been doing for brain cancer. And I'm going to go back to this example of the chaos versus rule model of cancer cell plasticity. And I picked this at the very beginning because the poster child brain cancer for the chaos model is glioblastoma, or GB for short. And GB is really one of the worst cancers, right? It's incurable. It's the most common malignant brain tumor. When you get it, it's more of a death sentence, right? 95% of the people that get it will live just a little over a year. And unfortunately, the first line standard of care, which is, temozolomide, which is a dirty, simple old chemo, and radiotherapy has not changed since 2005. And that's not because the community is not trying. Every single omic tool that you could think of has been applied to GBM. But what's been thought to be, again, the limiting step in these tumors is the high levels of heterogeneity and plasticity of the cancer cells. And really, summarizing a lot of amazing work from almost 20 years of work from lots of different labs here, what we know from all these different studies in that in any given patient's tumor, first, cancer cells in GB exist across the spectrum of different states. On one end, they resemble corrupted versions of normal human brain development cell types. They look like messed up oligodendrocyte progenitors or fetal neural progenitors or astrocytes. On the other hand, they look like states that normally don't belong in the brain, that are described as mesenchymal-like, for example. Then a bunch of, PDX studies or, transplantation studies of these cells into animal models have suggested that cancer cells have unrestrained plasticity. So if you take an oligodendrocyte progenitor-like cancer cell state, it's able to transition to any one of these other types of states. And then finally, to make things even more complicated, it's thought that the combination of tumor genetics and microenvironmental cues basically, promote the dominance of one of these given states in each patient. So if you have a lot of cells that are mesenchymal-like in their nature, then you have a tumor subtypes which is mesenchymal-like. So again, right, a lot of heterogeneity. That could be the problem. But going back into the, the result, the reasons I listed at the very beginning, we thought that the real challenge here is we don't really understand the fundamental biology, human biology of these cancer cell phenotypes. We don't really know what these cancer cell states mean. What does it mean to be mesenchymal-like in a tumor? We don't understand how they come to be. We don't understand how they transition from one state to another. And finally, we are not very good at predicting their plasticity. We have all the different omic data sets, but we can't use that to say, I think this cell state is going to become A, B, and C. So about 3 years ago, as part of this really wonderful, rewarding, Welcome Leap program that Shankar was also a big part of, I put together a bunch of awesome group of, friends and collaborators, a small international consortium, to address these questions by creating the most comprehensive multimodal atlas of human GB. I'll acknowledge people along the way, but I'll tell you that I think one strength we had is that except our wonderful clinician, researcher, Richard Mayer, the neurosurgeon who collected all the samples, none of these people are standard brain tumor people. So I think that enabled us to bring kind of a fresh perspective into this field. Right. So heart of this data set, or heart of this effort, is this amazing multimodal cell atlas that Tanya and Fani, senior staff scientists in my lab, commanded a little army of research assistants to generate. It's not the first cell atlas of GBM, but it combines two really unique features. First, it's really deep per tumor. So we look into just 12 tumors, but we look into each tumor incredibly deeply. We know that these tumors could be quite regionally diverse, so there's intra-tumor heterogeneity. To be able to capture that, we look at least 4 different sites of each tumor, and on top of it, we collect lots and lots of different cells and tissue sections from each tumor. We have almost 100,000 cells from just individual tumors, so that allows us to do really deep analysis. Second, to be able to put together the strengths of all these different modalities and account for the genetic heterogeneity of this cancer, we actually took the expensive solution and generated all these different modalities: single nuclei joint transcriptomics, chromosome accessibility sequencing, Visium, Xenium, spatial transcriptomics, and a bunch of other stuff here from every single sample from consecutive sections. So when we integrate stuff, we can take the single cell data of that tumor and map it to the match Visium data of that, right? So we got super true matching. And putting all this together with, new computational tools. We're really interested in all these different elements of GB biology. And today, just in the interest of time, I'll tell you a bit about the spatial tumor organization, the state trajectories. If you have time, we'll also go into the gene regulatory networks. Right. So as you saw before, we start with the single nucleus data, right? We use that true single cell resolution data to go about mapping the different flavors of malignant and the tumor microenvironment cell types. So my graduate student, Grant, spent a lot of time cleaning this up, and he created really this wonderful atlas over 1 million nuclei, profiled in QCs, both for the transcriptomes and ATAC-based chromatin accessibility. And as you can see here, we have all the different malignant cell states that we'll go into detail. But beyond that, from every single patient, we have also captured every single tumor microenvironment subtype, from myeloid cell types to neurons to oligodendrocytes to vasculature, so on and so forth. When we looked into the single nuclei data, to make a long story short, we found that tumor cells sort of varied across 4 different classes of substates, right? So a big group of sort of the first 2 groups look like these developmental-like cancer states. They resembled either oligodendrocyte progenitors, or cryptically, they also resembled neuronal-like progenitors. This is a glial tumor, but you can find these cancer cell states that really look like immature neurons that we discussed with Shankar's group earlier. So that's one side. On the other side, a big group included cell states that were previously described as mesenchymal-like, but when we looked into them in detail, we saw that they really resembled almost a transcriptional spectrum of astrocyte-like cells in one end. In the middle, astrocytes expressing an injury response and wound healing signature that we describe as gliosis or reactive gliosis in the brain, which is a brain injury basically state. On the other end, hypoxia. Right? And then finally, there are cells that were very clearly proliferative. The next question we had here was to see whether these cell states showed any spatial architecture, right? It was pretty, sensible at the time that maybe there's no architecture. All these different cell states are maybe randomly admixed in these tumors. However, when we integrated the single nuclei data with Visium, we found that there is exquisite spatial architecture to every single tumor looked at. And the rough pattern we saw here was that these developmental-like cancer cell states were mainly located in the periphery of these tumors that is bordering the normal infiltrated brain that the cancer is extensively or continuously growing into. But then as you sort of went deeper into the necrotic and the hypoxic heart of these tumors, you start seeing these zones of glial injury, gliosis dominated as well as hypoxia dominated tumor cell states. And since spatial transcriptomics data, right, is not just a pretty picture, we could quantify it. If I were to take all the data, almost 100 sections, tissue sections that we profiled from 12 tumors, and identify that sort of tumor tissue niches, sort of the spatial domains of co-located cancer and tumor microenvironment cell states, and show you a graph of their physical proximity to one another, where the nodes here are those tissue niches, and the connections, the edges, the thicker they are, the closer those 2 niches are in the physical space in spatial transcriptomics. We saw really the singular anatomical axis. Again, on the infiltrated brain side, where the tumors are expanding to, you're seeing those developmental-like cancer cell states. And as you kind of go deeper and deeper into the heart of tumors, you're seeing these stepwise spatial transitions into gliosis and hypoxia. And again, as a developmental biologist, when I see a pattern like this, and I know that cancer cells have the ability to transition across these states, that makes me think that perhaps this is a stereotype trajectory. Perhaps we are seeing this because as the tumors are expanding and growing into the normal brain, cancer cell states are progressively transitioning from these developmental states into injury response and hypoxia. So we could actually see many signs of this, right? If we looked into the gene expression signatures of cancer cells in these gliosis and hypoxia domains, you could really see kind of waves of expression almost, right? Cancer cells upregulating a lot of like integrins, serpins, annexins, these are like telltale signs of injury response and wound healing. But we wanted to be able to take a much more unbiased look at these trajectories and see if we could predict them from single-cell transcriptomics. So to do this, we did 2 things. First, we did something that is absolutely necessary if you want to look into cell trajectories in cancer data. We accounted for the genetic heterogeneity of cancer cells within each tumor. And in GBM and many other tumors, we know that across tumor development, cancer cells segregate into the genetically distinct phylogenetic subclonal lineages. So we are able to infer those subclonal lineages from joint single nuclear RNA and ATAC data. And what that showed us is that in every single subclone, these developmental and gliosis hypoxia states were co-located with one another, which makes it feasible that transition across these states. But after that, what we did was we grabbed each subclone, the cancer cells that make up each subclone, and you're seeing here one tumor, 3 subclones that make up this tumor, and we applied to this an unbiased trajectory inference tool that we created with Oli Stegel's lab. Student Alexander did this, that we call self-to-fate. So I'm very happy to talk about this, but in the interest of time, it's a tool that's a probabilistic model of RNA velocity dynamics. So it's like a biophysically granted tool that allows you to infer directional trajectories, meaning that you can sort of predict a beginning, a middle, and an end. It's not just a pseudo time that can go both ways. And what we saw here in stream graphs that resemble the distribution of these different cancer cell states is that no matter which flavor of developmental like cell state was in each of these subclones, they all had the same thing in common, a progressive transition from developmental progenitor-like cancer cell states into gliosis and hypoxia. And finally, when we expand this analysis to every single subclone from every single tumor, we saw that no matter which cancer cell state these tumors were enriched for, what their tumor subtypes were, they had the same thing in common, progressive stereotype trajectory. So tying all that together, our new model for this is that heterogeneity in these tumors actually dates back or can be traced to a fairly simple stereotype trajectory of cancer cells, which again, I say this 7 times in this talk, that starts with developmental-like states at the site of tumor expansion and progressively turns into a glial injury response and hypoxia. And this trajectory is actually spatially patterned in the tumors. And it's something that shows up in every single tumor that you can look at, and it shows up in every single genetic lineage, sublineage of tumor. So we're obviously very biased, but we like this model first because we think, A, beyond just calling these states mesenchymal-like, neural-like, this-like, I think we have a biologically inspired model here, right, which is much more interpretable, explainable. I think we have cleaned up a couple things with regards to tumor subtype classifications. We think that a lot of studies that are just looking at single biopsies, single sites of these tumors might be massively confounded, because if you kind of profile just the top of this tumor, you'll completely miss the gliosis and the hypoxia site. But moreover, I think the thing that I'm most excited about here is that we've been able to take this chaotic model of tumor biology and come up with a tractable cellular trajectory of cancer cells that can be pinpointed in every single patient, which I hope will be the beginning, the foundation of our efforts to develop new targets, targetable, like mechanisms, disease mechanisms in this cancer. So in the next 2 minutes, because I'm going to go over time, I want to talk about some of the orthogonal work we did in the prediction space, right? So we had a lot of data, and a big part of this data was the single nuclei multi-omics, where we co-profile the transcriptomes and the ATAC-based chromatin accessibility of each cell. We did this initially because we wanted to leverage this data modality to identify the gene regulatory networks driving different cell states, right? So be able to do this shared student between Oli and Moritz Small's lab in DKFZ, Manu, put together a completely new deep learning framework that we call SC-DORI. And what SC-DORI is, in the interest of time, I won't go into details, really is taking this sort of standard in silico chip-seq approach, right? I have a transcription factor. It has a predicted motif. Can we look for the presence of this motif marked by a tag signal, and does the downstream gene express? But what it does is that it really maps that into a clever, inference framework that allows you to infer different gene regulatory networks in a given data set. And then you can actually compare the abundances of gene regulatory networks in different individual single cells. So in a way, you could think of it as almost like a dimensionality reduction for GRN analysis. We're not just mapping one giant gene regulatory network. We are saying the regulons of cells consist of different abundances of these different types of gene regulatory networks. And what that really enables you to do is you can go into your data at single cell resolution and say that cancer cell state A is gene regulatory networks, 1, 2, 3, and cancer cell state B is gene regulatory networks 4, 5, 6. Again, when you do this, you can get this kind of stuff, right? You can say, here are my different cell states. Here are all the different transcription factors, candidate regulators of this. But again, since we're doing this at single cell resolution, what we realized here is that we can infer cell state transitions from their differential regulation. So we could simply ask, do the transcription factors enriched in cell state A, are they predicted to activate or infer the transcription factors of cell state B? And we could do this in a pairwise manner across all the different gene regulatory networks identified in the data set. And what we can do that with, that type of comparison, is to synthesize it into basically a cell transition, a predicted cell transition score. So here what you're seeing is we got our UMAP of malignant cells. The 4 different major flavors are marked as the nodes, and the arrows or the inhibition marks literally show you the quantified cell state transition or the inhibition potential across these cell states. And when you look into this, what you're seeing is not everything connected to everything else. You're actually seeing that, for example, OPC and PC-like cancer cell states have a high probability of becoming neuronal-like or astrocyte-like, but they have a fairly low probability of turning into gliosis and hypoxia. And what you also see here is that once cancer cells transition from being astrocyte-like into gliosis and hypoxia, they have a predicted low potential of coming back. So in other works, from gene regulatory networks, we're also predicting a hierarchical cancer cell state transitions. In this paper, there's a lot about this really interesting angle, which is you have a cancer cell state that actually is enriched for repressors that are keeping this guy from becoming or straying away from a neuronal-like cancer cell state. And we did, Morris's lab did a lot of functional analysis on this, and we're expanding on this now with more analysis. So if you're interested, please do check that out. Okay, as I close, I'll also just say that when we generated this, we wanted these data sets to be reused by others, right? So even with the preprints, what we managed to do was to allow you to download and interactively explore all of the single cell and the spatial transcriptomic data that make up GBM-space. For this, a wonderful IT team in our program adapted the Web Atlas framework we published earlier this year. And again, if you're interested to download the data, play with it, or just sort of navigate the spatial and the single cell data in a sort of just based on the web browser without downloading it, please check out the portal. Okay, so as I close, I hope my takeaway to you is that using this type of multimodal cell atlasing and biologically inspired analysis, we were able to go from this model of no rules to a fairly stereotyped setting of rules, which I hope underpin, again, the pathology of these tumors, which again, I hope will be beginning of a new era, at least for us, to devise new therapies against it. So I'll finish by, again, acknowledging the wonderful individuals that have been in the lab over the years. I think, both on the autism side, but on the GBM side, I think it's been really a huge honor to work with all of these people, but also our very long list of collaborators and our funders, specifically Simons and Wellcome Leap for the autism and the GBM projects. So with that, thank you so much for your attention. And if there's time, very happy to take questions.
Well, absolutely outstanding.
Wow.
Thank you. Thank you very much. And so let's open it up for questions, please. Yeah.
I was wondering if you could talk about what you see as best practices for collaboration with computational groups.
Yeah. No two computational groups are the same, right? I think they're like different flavors, but I'll tell you that I think to me, the key has been like a back and forth and like a shared partnership. So, I started out as a just wet lab biologist, and you're never gonna sort of get the best advice from me on probabilistic modeling parameters. So I knew that if you wanted to go into this space and to address the type of biological questions we've been able to address with cell to location, cell to fate, we need a collaborator like Oli. And the relationship that I at least have with Oli is like a back and forth. So I respect his biological understanding of the problem that we're working with. He respects my appreciation of the computational bottlenecks in this and the modeling parameters. And really, I think it's also, I think, a lot better if you have people on board from the beginning of the project rather than after you generate your data and bring on someone else. And I think last, I think, you know, I wish I had access to some of these opportunities when I was training. Cross-training, I think, is extremely important. I really think that the new generation of students in the postdoc that we're training should be able to do both. It's not a failure to just do one, to excel in one, but at least to be able to speak or understand a language of computational biology, I think will allow you to make so much more progress as a wet lab scientist, even if you're just better at managing that collaboration. Yeah.
Absolutely beautiful talk.
Thank you so much.
To see you using multi-modal technology to understand such a complicated, both spatial and also temporal complication of GBM. I was wondering how much of the information you got independently from your spatial technology.
Yeah.
You know, how much you would need to rely on combination of the two. Sequencing, the single cell sequencing, and also the spatial.
Yeah. It's a great question. And the more we've done this, especially for GBM, the more it became difficult for me to sort of pick a favorite, right? So I'll tell you that when we did this cell typing in single nuclei, which is not spatially aware, just be looking at this single nuclei, we saw this and it made us think that at least in this astrocytic gliosis and hypoxia, you're not seeing it here, but there's a lot of genes that seem to be in gradients, right? We have a figure on that just in the paper. But it's not until we saw the spatial transcriptomics data, this really kind of like organized stepwise transitions across these cell states, that we actually started thinking about the possibility of stereotype cell state transitions. So I think that was, to me at least, really, when I was saying at the beginning, biology is spatial, this was the example I'm thinking of. If we did not have this finding, we would not have formulated a hypothesis to test the trajectories in the single cell data of a trajectory inference. And when we did this trajectory inference, I don't show you this, but all these different genetic subclones, for example, are initially mapped by copy number inference from the joint RNA and the ATAC data, which we also did in the paper using copy number events detected at the DNA level, validated by spatial DNA sequencing of the same tumors. And if we didn't have that, I think, again, the temporal trajectory picture we would have extracted from single nuclei would have been super complete. So I'm looking forward to, I hope there will be a time in the future, right? We'll be able to spatially measure full transcriptomes and full chromatin accessibility in the tissue with a single technique. But we're not quite there yet, and I think we just need to leverage as much as we can from the integrated methods.
And so I have two questions, one that's kind of specific and one that's kind of big picture. So you're talking about SC Dory,
right?
And then you talked about that the ASD work in the thalamus.
Yeah.
Where you did xenium and you looked at those 250 marker genes for ASD. But you also have the single cell data for the thalamus.
Right.
Have you applied SC Dory to see if, because
right.
a lot of the mark, you know, like TF7L2, that's the transcriptional regulator. So have you?
Yes, I like this
Can you go back into that? I don't know if you did this in paper, right? But you know, for like TCF7, does SC Dory predict that in the thalamus from the multiome data is predicted as a key regulator, or are there other ones that are perhaps like novel ASD markers that you could extract by integrating those methods? That aren't in a list of 250 because nobody's done what you did.
No, it's a great question. We haven't done that. Primarily because we actually don't have single cell data from developing human brain. The UMAP I showed you, you believe it or not, was actually just from the xenium. And I think in xenium, it's kind of, I think the regional expression patterns are so strong that the embedding looks almost like single cell transcriptomics. If you're familiar with xenium or Murfish embeddings, often the boundaries are mushier. That's because there's bleed through of genes between cell types and stuff measured in space. I think what you're saying, Andrew, completely makes sense. A bunch of these TCF, let's forget the name of the gene, TCF 7L2, very well-known thalamic sulfate regulator, post-mitotic regulator. I didn't show you guys that, but if you dig into the germinal zones, you see this huge enrichment of transcriptional regulators. So I think there is an awesome opportunity to think about now, okay, how does really what is the downstream mechanisms of, when you lose transcription factor A, or when you have haploinsufficiency specifically, or more accurately, of transcription factor A, what do you get in the germinal zones? What I will implore, though, in this, as we think about how to approach this functionally and follow this up functionally, you want to go, your hand wants to go and write the grand proposal where we say we knock it out in organoids, right? We CRISPR it and this and that in cortical organoids. But I think we still need to think about what is actually the most physiologically relevant model system for that. And if we thought, and we pitched assignments, and they did not like that, unfortunately, but we'll try again. We thought for this, a really good model would actually be a haploinsufficient mouse, right? Because a person who has a rare autism risk variant does not have it magically just in the germinal zone progenitors. It has it throughout its entire body. And it's just one copy of the gene that has the loss of heterozygosity, right? The other one. So I think in terms of how we translate this into functional output after the predictions, there's still some work we have to do, but we're quite interested in it. And if you guys are interested in that kind of stuff, let's talk, because we love that.
Yeah.
Okay, so my second question is, so you're kind of at this interface of neurodevelopment, cancer, neurodegeneration. Do you feel like as you've gone more into studying those different niches, that the lines get blurred between them of like,
yeah.
is, because I think we have like this key demarcation of, well, there's cancer,
Yeah.
and then there's neurodegeneration.
Yeah.
Those are two different things.
Yeah.
Where does is there brain?
Yeah.
Right.
So I'll tell you, the cost of trying to study as a new lab, or we were once a new lab, now, no longer qualify as an early investigator, but I think the cost of doing this is that, right, you spread a bit too thin. I think if we drop one of these diseases and just like focus just on GBM, we would have done it faster. That's there. But I think the benefits of it are tremendous. And I can tell you, we have actually we have accumulated so much biological perspective from studying different diseases that allowed us to apply not just the technical stuff and tricks, but also the biology of it. So when we think about wound healing, and when we look at neurodegeneration, it's really amazing to see. It's like a lot of the same machinery. And I think a really awesome historic analogy to this is that, this is an actually question to the audience. So how many genes are in the human genome? How many people what do you think people predicted? What do you think was the number of genes people expected to be in the human genome before we did the sequencing?
A lot more.
A lot more. People thought that there are 100,000 genes, 200,000 genes, and we ended up at 30,000 genes. And that just shows you that the same list of parts is used in these variable but recurring combinations. And I think that, to me, is the most astounding sort of like deliverable of all these omic data sets. And I don't think we quite have that sort of complexity mapped, or we can't quite interpret that. I hope that we have is that if you interpret that right, whatever drug you develop for neurodegeneration can be used to repurpose against some aspect of brain cancer. But again, I really share, I think, also your optimism in that I think we'll be able to do that.
Can you ask the people in Zoom have
any
questions?
No,
I'm not sure if they could hear us. I hope they could.
there are no questions.
No questions.
I do have a question. So
Sure.
it's very exciting work. So what would it take to couple all this with physiology or doing activity measurements at the same time?
Yeah.
For instance, you showed this great coupling, right? There's thalamus and there's cortex.
Yeah.
Thalamocortical loops. And, this very much involved in that.
Yeah.
Right? It's all brain waves are associated.
Yeah. I think for the autism part, I think it's a simple answer. a mouse, right? Like a functional mouse. Like, again, I just want to say, let's do like a thalamocortical organoid model. And I think there's a lot of promise to that. I think we should, right? And I think there's like in assembloids, there's been incredible progress. And you could do fun stuff. You could just knock out the genes in the thalamus and have them hook up to a normal cortex, right? You can do fun stuff like that. But I think, as you would appreciate, I think it's incredibly important that we have a system that is capable of behavior. So what I would love to do is to literally grab every single one of these thalamus enriched expression enriched variants, make a haploinsufficient mouse for every single one, and profile the heck out of them. So longitudinally profile with single cell spatial omics, how thalamic development looks like, and then functionally profile at the level of connectomics, but also behavior. And in thalamocortical loops, there's just really beautiful, right? Like higher order, first order, like lower order stuff that you can do, lots of beautiful functional phenotypes. In GBM, our answer to this is that a lot of the functional testing, I think, should be done in more human models. And the model that we love, that I'm happy to talk about more, is patient-derived organoids. So not a normal brain organoid where you're knocking in the GBM mutations, but literally grabbing a piece of tumor that we normally put into the 10x machine and maintaining that as an organotypic culture situation. And we have a lot of data, we and others have a lot of data showing that they are capable, those systems are capable of recapitulating or maintaining the regional and molecular identity of the parental tumors.
That's very exciting. Yeah, I have a question, but you have a question, but
Yeah, so you mentioned that the grad student made an atlas with cleaning the data. So I presume their data cleaning had something to do with all of the results were cleaned.
Yeah.
Did you have any special cleaning techniques in the lab? B, did you find very different results when you clean things?
Yeah. It's a super important question that we never talk about, right, in presentations. I think the most important thing, our number one data cleaning device, are Tanya and Fani that you're seeing here. It's like garbage in, garbage out, right? I think when we do these nuclei extractions, we take a lot of care to extract nuclei in a happy way. And, in these brain tumors, cancer tissue, you got all kinds of necrosis, this and that going on. So being careful was really important. Second, I think we don't use anything special. I'll say we are often stringent, right? So the QC parameters that we use for the RNA or chromatin accessibility, which we all document if you're interested, happy to point at them. We tend to pick stringent ones. So I think the total number of nuclei we sequenced was probably closer to 1.5 million, and we threw probably 1.3, 1.4 million. We threw away quite a bit. There are some papers where the number of nuclei we throw away would be the atlas for that paper, right? But I think it's quite important because if you think about it, we have spent months and months just analyzing that data set. So again, if you took shortcuts at the beginning, you'll pay for them down the line.
Yeah. Noise, more noise there.
Yeah.
What happens in your beautiful trajectory map? What happens to the synapse?
Yeah, it's really, really interesting because so number one, I didn't share that, but so you see here, we have these two flavors of developmental-like progenitors. The one that looks like neuronal-like is really like legitimately neuronal-like. It looks, to be more accurate, it looks more like a double cortin positive immature neuroblast, but you see so much synaptic gene expression that you don't see in the other cell states, right? So that's very cool. Number two, the subclones that I'm showing you here, 8010, 0, 1, 2, the three subclones, the one that's enriched for the neuronal-like cells, last one, is very clearly the late tumor subclone, meaning that it's marked by a very distinct copy number event, a chromosome 17 alteration that you don't see in the first tumor subclones. They all have the diagnostic GBM mutations, chromosome 7 and 10. So what actually happened here to us is a super cool thing we want to exploit and find more examples of. What happened here is that during late tumor development, a genomic alteration within the tumor gave rise to a new cell state or promoted a new cell state. And to maybe spoil some of the work we're doing now, what we think is possible to identify regulators of these cell states is to do this more and more and more, both at the level of inferred copy number events, but also in substitutions and mutations and such, ideally with single-cell DNA sequencing down the line, and find enough examples of, in this tumor, this was cell state A, this mutation happened, now we have cell state B. And I think there's going to be quite a room for exploration there with that kind of approach.
I mean, it ties back to Bert's question on whether you can map synaptic density across space.
Yes.
Right, using, I mean, your projected pseudo trajectory.
Yeah.
Space.
Yeah, and you can sort of see here, it gets a bit sort of like murky in that last subclone, the synaptic density will be super high early on, right? Then gets lower and lower.
Exactly.
But also there's some cool stuff going on with OPCs that are actually hooked up into the synaptic circuits in the brain. So the field of cancer neuroscience, I'd actually say the major cell type, cancer cell state that concerns that, as far as we know now, is these OPC-like cancer cells. And they express only parts of the postsynaptic machinery, but not the pre.
Right.
Yeah.
Right. The question really is also that you can stimulate these cells to change the trajectory of the tumor.
Yeah.
With a functional connectivity.
Yeah. Okay, one last question.
Yeah, what is the effect in this case of the synapses or the glia when they have the tumor? What's happening in this case? There's some correlation with the extension of the tumor over the glia or something, because you have happen the effect in the synapses. Right?
You mean, yeah, so what is, you mean, are you saying what is the kind of like the, what is the consequence of having the tumor connected to the normal brain circuits, to synapse with normal brain circuits? Is that what you're asking, or different?
You have the tumor.
Yeah.
And they affect the synapses.
Yes, okay.
But the
Okay.
synapses, the effect in the synapses is there are some relation with the glia.
Yeah. So I think it's a two-step. So your best guess is as good as ours on this, but I'll tell you, like, the field of cancer neuroscience, which is really trying to understand the electrical communication between tumors and, the nervous system, is based on this. And the idea is, and in GBM, I think there's very good now imaging and some level of recording evidence for this, is that, brain tumor patients often will have personality changes. So it's like character changes and stuff. It sounds like a Hollywood movie device, but it happens. People suddenly have a completely new personality and stuff. And a part of that, so that's the good bit, but on the worst side, they can have epileptic seizures and stuff, and they have tumors. And all of that is happening before the tumors, because the tumors are actually synapsing and manipulating normal cortical circuits. That's number one. Number two, it's thought that the glial cells in those activated neural circuits are acting accordingly. So there's thought to be increased myelination of those regions that are hyperactivated, because it's thought that activated neurons signal to OPCs to proliferate and blah, 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 blah. But also it's thought that there is a complex cascade of glial injury response outside the tumors. And there's some really cool work showing that as the tumors are growing, at least in mouse models, they actually receive signals from dying neurons and normal glial cells, and they almost like co-opt those cues to support their progression. So I think it's this beautiful crosstalk, complex crosstalk going on, basically.
Yeah, because they change they interchange the neural needle between the glia and the
Right.
Thank you.
Thanks so much. Yeah.
Fantastic. Let's thank our speaker again.
Thanks so much. Yeah. Thanks so much. Yeah.
Thank you very much. You can catch up with Omer if you have other questions, and then you can cross.